Biochemical tests reducing sugars, non-reducing sugars, starch, lipid. Proteins. Reducing sugars. Test. Add 2 cm of a solution of the reducing sugar to a test tube. Add an equal volume of Benedict's solution. Shake and bring gently to the boil, shaking continuously to minimize spitting. Observations. The initial blue coloration of the mixture turns green, then yellowish and may finally form a brick red precipitate. Basis of test. Benedict's solution contains copper sulfate. Reducing sugars reduce soluble blue copper sulfate, containing copper L alliance to insoluble red-brown copper oxide containing copper LL. The latter is seen as a precipitate. Non-reducing sugars test. Add 2 cm of sucrose solution to a test tube. Add 1 cm dilute hydrochloric acid. Boil for 1 minute. Carefully neutralize with sodium hydrogen carbonate, check with pH paper. Care is required because effervescence occurs. Carry out Benedict's test. Basis of test. A disaccharide can be hydrolyzed to its monosaccharide constituents by boiling with dilute hydrochloric acid. Sucrose is hydrolyzed to glucose and fructose, both of which are reducing sugars and give the reducing sugar results with the Benedict's test. Observations. As Benedict's test. Starch test, iodine/potassium iodide test. Add 2 cm 1% starch solution to a test tube. Add a few drops of L2/Kl solution. Alternatively, add the latter to the solid form of starch. Observations. A blue-black coloration. Basis of test. A polyamide complex is formed with starch. Lipid. Emulsion test. Add 2 cm fat or oil to a test tube containing 2 cm of absolute ethanol. Dissolve the lipid by shaking vigorously. Add equal volume of cold water. Basis of test. Lipids are immiscible with water. Adding water to a solution of the lipid and alcohol results in an emulsion of tiny lipid droplets in the water which reflect light and give a white, opalescent appearance. Observations. A cloudy white suspension. Protein. Biochemical test. Add 2 cm protein solution to a test tube. Add an equal volume of 5% potassium hydroxide solution and mix. Add 2 drops of 1% copper sulfate solution and mix. No heating is required. Observations. A mauve or purple color develops slowly. Basis of test. A test for peptide bonds. In the presence of dilute copper sulfate in alkaline solution, nitrogen atoms in the peptide chain form a purple complex with copper L alliance. Biuret is a compound derived from urea which also contains the con group and gives a positive result.